All right, everyone. So my name is Cristina Gomes Gonzalez. Some of you I know from working with you on the research studies that we've been doing together. Other of you might not know me. So I'm the Director of Program Operations here at Vanguard, and I've been working in research for about 20 years. So today I'm going to talk about sort of protocol deviations, what they are, what we need to do for them, and just to kind of give you guys a better understanding of this. Slides are not. Oh, okay. So the goals for today's presentation is I want to give you guys a deeper understanding of what a protocol deviation is. We want to talk a little bit about how to assess their severity and their reportability to the IRB. We're going to talk a little bit about how to properly document these deviations, and then how to create kind of a corrective and preventative action plan so that we don't have deviations happening all the time. Having said that, you know, protocol deviations happen in research. They're inevitable. No one is perfect, and we all make mistakes. But what's important here is sort of how we manage those and how we make sure that we're emphasizing them and addressing them so that these mistakes don't keep reoccurring. Because some deviations may be minor, but as they occur more frequently, they could potentially be moved to sort of major deviations. So let's start off with the definition. What is a protocol deviation? So the ICH definition is that a protocol deviation is any change or divergence or departure from study design or procedures defined in the protocol. A point I want to make here, when I started doing research, to me, this was very specific to the study protocol, right? And as many of you know, most protocols don't really go into good clinical practice or the guidelines and regulations that are set forth in research. And so sometimes we will have deviations that sort of deviate from our good clinical practice and regulations that have been defined to protect the safety of patients, as well as to ensure the integrity of the data. So sometimes a deviation could be, you know, an error in the consenting process. Now, obviously, that's not clearly defined in a protocol, but it is still considered a deviation that we would need to sort of address. And I'll go into a little bit more what some of those look like in the later in the presentation. So a deviation is actually also referred to in different ways, depending on the IRB or the agency you're working with. So some terms to be aware of is sometimes they're called protocol violations, sometimes they're called variances, sometimes it's non-compliance, and then sometimes it's called a change in research. And this mostly refers to maybe planned deviations. An example of this is something like, you know, when we had COVID, a lot of our research sort of changed, and we had to make some plans on how we're going to continue doing research within the COVID environment. So that's sort of a planned deviation that you would report to the IRB and say, this is how we plan to sort of mitigate the COVID challenges we're having. Some of the more common deviations that we see in research is informed consent procedure problems. So this can vary from not having the patient sign the consent form, not initialing in the right places, signing the wrong version of the consent form if there has been a recent change. And so these are, again, not listed in your protocol, but they're important to sort of look at and identify sort of what happened and how we can fix those from reoccurring. Some of them could be just administrative and minor, but some of them would be sort of major, which I'll talk a little bit later about what that means. But many times we do see problems with the informed consent procedures. And this is the very first place where a patient sort of interacts with the study, and we want to make sure that they are informed and that we've documented this informed consent process correctly. Next are problems with the inclusion-exclusion criteria. And this refers to both patients not meeting the criteria, but also just proper documentation of these. So you want to make sure that in your source that all the inclusion exclusion criteria has been addressed, and that there's proper documentation that the patient does meet that criteria for the study. Next are safety assessments. So what I'm referring to there are maybe labs that were missed, or labs that the patient, I guess, was not able to complete because they are safety assessments. And it's not just labs, we're talking about maybe vital signs, maybe we're talking about ECGs, depends on the protocol and what's sort of considered a safety assessment. But if these are missed, they could put the patient potentially at risk or in, harm's way, and we want to try and avoid those. So these are definitely deviations to make sure you pay close attention to. Other common deviations are things like study procedures being missed, maybe a small assessment or out of window assessments. Those are small deviations that sometimes are just administrative, but depending on the importance of that procedure or that assessment, it could be considered major, especially if it affects kind of the primary endpoint of the study. Then we have adverse events, so documentation and reporting those. So adverse events are really important in research because, again, we're talking to the safety of the patient in the research study. So these are important to sort of assess at every visit, make sure you're documenting. Sometimes what will happen is a CRA will come on site to monitor your site, may see that in the progress note there's an AE that you may have forgot to document in your source. And so that could potentially become a deviation because there are timelines for reporting these. And if they become a serious adverse event, then there's also IRB reporting that we would need to do. So just making sure we're documenting those and reporting those procedures. Any deviation to that obviously would be considered a protocol deviation. Next are randomization procedures. So some examples of that is, if you randomize a patient without having, checked all the inclusion and exclusion criteria at the baseline of visit, and that obviously would be, a deviation. If they didn't meet a certain criteria and you randomize them, that would be a problem. And then the last one I have here is study medication management. So there are various places here that we could have deviations. If a patient, takes too much medication, that's something that potentially would have to be reported. If a patient doesn't return the medication bottles when they were supposed to, could potentially also be a deviation. So again, this is really dependent on the protocol and what the protocol asks of how you manage this medication, but just some things to consider. This list is not all inclusive. There could be deviations in other things, but these are sort of the more common ones that you should look out for. Okay, so who's responsible for deviations? So the short answer here is that everyone, right? So anyone who's working on a research study should be looking out for any potential deviations or differences that are happening that should be reported or addressed in some way. So the research staff, obviously, they're the first line of interaction with the patient and the source and documenting things. So a study coordinator might be the first person who sort of identifies, oh, we did this visit out of window. But really, it could be anyone, the CRO, when they're looking at the data, vendors, when they're sort of transferring data to the sponsor, may find that there's a problem with the data, monitors, obviously, and then also the sponsor. Okay, so it really, anybody involved can actually identify one. Once a deviation is identified, sort of have to assess its reportability to the IRB. Is it considered sort of a minor or a major? Ultimately, the IRBs do leave it up to the PI as the responsible party for assessing reportability of the deviation. If you do have a deviation, you want to make sure that's discussed with your principal investigator, and that discussion is documented in the sense of, what was the outcome of the decision. Now, sponsors and CROs also play a part in this decision. So there are some deviations that maybe a sponsor or CRO will consider a major deviation, whereas maybe a principal investigator would consider minor. And so those discussions then would need to sort of happen. Again, this really is dependent on the protocol and on the procedures that the protocol states. And so if something doesn't really affect the primary endpoint, a sponsor may say, oh, this is minor. So it's sort of a collaborative discussion. If your principal investigator doesn't feel it needs to be reported or doesn't feel it's major, then you should have a discussion with your CRO and your sponsor about that. Okay, so how to assess the severity? So we've been talking about there's minor and major deviations, and there's some differences in how the IRB wants to see this reported. So the first thing to really understand is, what is your IRB's reporting criteria? Every IRB sort of has different expectations of what they want reported. So it really could vary from IRB to IRB. If you're using kind of a local IRB, then that would be your agency's kind of regulations that you would need to be aware of. For central IRBs, there's usually a manual out there that you can look at. Okay, what are they defining as a reportable deviation and how you report it? So some IRBs want all deviations, so the minor ones usually get reported with a continuing review, and then the major ones are usually reported right away. Other IRBs only want to see the major deviations. And again, I'll talk a little bit more about that a little bit later, but major deviation would be something that affects the subject's safety or the integrity of the data. Okay, the second thing you want to do is understand the protocol. How does this deviation impact the patient safety in the study? And how does it impact the study data? So does this deviation somehow alter the results or impact the results in some way where we can't have sort of a standard way to generalize the results at the end of the study? And a lot of times this refers to, does this impact the primary endpoints? So if a study is looking at maybe their primary endpoint being a rating on a certain assessment, let's for example say a PANSS assessment, then if your patient does not do that PANSS assessment at the end of study visit, then that would potentially be considered a major deviation, because now we can't use those results. We don't have the endpoint that we're looking for. Okay, and then lastly, you want your principal investigator to determine the severity. Is it a minor or is it a major deviation? And that sort of then helps you take the next step with the deviation. Okay, so to talk a little bit more about kind of what a minor and a major is, and sometimes these are referred to differently, like eminent, I think sometimes it's referred to, but I think the common wording is minor and major. So minor deviations usually are reported during a continuing review, and these are more administrative deviations that maybe they don't impact the patient safety. They also don't substantially impact the integrity of the data. And these, again, get submitted yearly with your continuing review. One thing to keep in mind, though, is with minor deviations, if you do have them reoccur a number of times, they could potentially then be considered a major deviation, which then you would be asked to report to your IRB. So something like you continuously use the wrong version of the ICF. If you do that more than 2 or 3 times, then that's something that would need to be reported to the IRB as sort of a major deviation that would need to be addressed. A major deviation, you do need to report as soon as you become aware of it. So either your monitor comes on site and says, oh, you have this deviation, you assess that it's major, it needs to be reported right away, okay? And when you're thinking about these, again, you're looking at, does this impact the safety of the patient in the study? And does this have an impact to the integrity of the data? And that usually is the IRB's definition of what to consider reportable, is looking at safety and looking at integrity of the data. Okay, now that we've identified, we've assessed. Now we need to make sure it's documented correctly. So at your site, you should have a study-specific protocol deviation log. Sometimes that's provided by your CRO. Other times it's expected that you have a log ready to sort of document all your deviations. So all deviations need to be reported on the log, either minor or major, they all need to be listed. A couple of other things you need to list on the log is you want to make sure that you have the date of occurrence, you have what patient it affected, what study visit, a clear description of the issue, what you did to correct the issue, and then your PI's determination of, is this a minor, is it major, is it something that we need to report or not? When your monitor comes on site, typically they'll look at these logs. If they find a deviation, they want to make sure that you've sort of addressed this on your log. So just being proactive on that will sort of avoid future work after your monitor comes on site. Okay, the other way to sort of document is if something is considered major and we need to report to the IRB. Usually the IRB has a reportable event or a deviation form that you would complete and submit to the IRB. And again, you want to do this as soon as the assessment of the deviation has been made and you guys have assessed it to be reportable. And then for minor ones, if it is applicable with your IRB, you would report that at the continuing review. Okay, and then you want to just document that the IRB has acknowledged that. So once you report a deviation, the IRB will send back either some follow-up questions like, okay, what's your corrective action plan here, if you hadn't submit that, or they'll just send you an email saying this has been acknowledged. And so you want to document on your log when that was acknowledged by the IRB. Okay. And then just a quick reminder here, again, that all the deviations should be documented as soon as they are identified. Okay, so if you think about sort of our ALCOA principles in GCP, we want to sort of follow those, right? Contemporaneous, original, accurate information. And that's sort of where all of these requirements come for your logs and reporting. Okay. Once you've sort of identified, assessed, documented, sort of want to think about how do we prevent this from happening again? And, again, we all make mistakes, especially in the first couple of patients that you may pass through in your study. You may not be 100% familiar with the protocol and you may miss some things, and that's totally understandable. But a couple of things to keep in mind. Make sure you consult the protocol frequently, especially in the beginning and as you're getting familiar with the protocol. Many sponsors do provide a mini protocol, and these are really helpful because you can carry them with you, right? And I sometimes try to flag the more important parts so I can pull to them, like the inclusion-exclusion criteria or safety sort of assessment and reporting criteria, maybe tables at the back that you need to sort of reference if there are certain criteria about maybe equivalency of medications or medications that are not allowed in the protocol. You don't sort of want to assume that because it's done in clinical practice that the protocol actually allows for it. So you always want to sort of frequently check that, and that'll help sort of reduce the number of deviations you have. Another important thing to do is as soon as a deviation is identified, you want to sort of address it right away. Some of them may seem very minor, but it's important to sort of figure out, what happened, why did we have this deviation? Do we need to provide retraining? And in a lot of cases, that would be one of your actions, is to sort of retrain the personnel involved in the deviation. And then sort of discuss it at a team meeting, even if it's minor, to make sure that you guys are addressing it so that it doesn't reoccur again. For more sort of major deviations, you want to make sure you're creating a corrective and preventative action plan. These are called CAPAs. I'll talk a little bit more about it on my next slide. But basically, this is a way to sort of go back and look at what happened and create kind of a procedure to try to avoid these from happening again. This could be informal, or in some cases it needs to be more formalized, but the discussion should happen with each deviation. In some cases, like I said, with the major deviations, the IRB does require to see a corrective action plan. So you'll have to sort of submit that to them before they acknowledge your deviation. And the other thing to keep in mind with corrective action plans is that you want to review them periodically. You may come up with a preventative action that maybe, 2 months down the line, you realize, oh, wait, that we didn't really get to the root cause of why this happened, and our preventative action is not working. And so you may have to sort of revise that preventative action to try and address the deviations from reoccurring. Okay, so what is a CAPA? So a CAPA really is sort of a deep dive into what happens with the deviation. And you want to ensure that you have kind of a thorough knowledge of the steps that are involved in whatever deviation happened. Maybe an example here is if your labs come back as hemolyzed pretty often, and that's something that's sort of common. You sort of want to look at, okay, how are we storing the labs before we ship them? How are we spinning them? Kind of what steps go into the process where you could have had an error, and try to pinpoint where that error is happening so you can correct that. So some of the steps here is obviously identify the issue, have a clear understanding. You want to ask questions to try and pinpoint to the root cause. A lot of times this is called a root cause analysis. And one technique that we like to use is sort of ask yourself 5 whys. So why did this happen? And then depending on the answer there, okay, why could have that happened? And so as you asking yourself the whys, you could get down to, maybe there's a problem with the FedEx pickup, schedule where your labs are always being picked up at the end of the day, which is causing them to sit there all day and maybe become hemolyzed. Might not be anything you did wrong in that situation, but maybe it's an outside process or procedure that you need to look at to be able to fix. Then create the corrective action. So in this situation, maybe you will redo the labs for the patient if your sponsor approves that. And then you document, your actions. So you redid the labs, maybe you provided retraining, maybe you asked your FedEx person to pick up earlier in the day. Maybe your team decided to call FedEx and schedule a pickup instead of waiting for your regular FedEx person to pick up. So once you've identified sort of what you did to correct that action, you document it, then you want to make sure you have a procedure in place to sort of prevent that from reoccurring. Okay, and again, that might be, calling FedEx to pick up at a certain time instead of waiting for your FedEx person to come along. So those are CAPAs, and we do have a template if you guys ever need something that we could share with you. Yeah, that's sort of the end of the presentation here. Does anyone have any questions?
Yeah. No, thank you.
Excellent. Well, we are available anytime you guys need us. Just send your project manager or Vanguard Research Group an email if you need any help. And thank you all for joining.
Thank you so much.